dear students. Today we'll discuss physiology of nerve fiber. It is as per NMC competency PY 3.2. Describe the types, functions and properties of nerve fiber. So objectives of today's talk is types of nerve fibers, functions and their properties. So let's start. Human CNS contains about 100 billion neurons and 2 to 10 times this number of glial cells. These are basically the supporting cells. So basically CNS is nothing but basically combination of neurons and glial cells. This combination forms the brain, spinal cord, nerve, everything, whatever. So basically two things are there on the CNS, neurons and glial cells. It has been calculated that 40% of human genes participate at least to a degree in formation of CNS. Now the most basic unit, neuron, let's have a study of brief regarding what is a neuron. So if you see the structure of the neuron, it has got important parts. Main part, the cell body. This is the cell body. The dendrites, this is called as the axon, and this is the synaptic connection, means it is a connection between two neurons. So a neuron is nothing but combination of this. Now the length of this axon varies with its placement where it is present. It may be very short, it may be very long leading to formation of nerve or tracts, fasciculus, and very short, for example, in basal ganglions with small communication. So basically these are the only key parts. So to close look to this one, you can see a typical cell body is there, and the neuron is nothing but typical cell having all this contents of a typical cell like nucleus, nucleolus. It will have the mitochondria, endoplasmic reticulum. So just like any cell of the body, there is a production of neurotransmitters here. So it has got same contents of any other typical cell but specialized with formation of neurotransmitter. Now based on the presence of a neurotransmitter, they are labeled differently. A neuron which is forming an inhibitory neurotransmitter is called as inhibitory neuron. A neuron which releases excitatory neurotransmitter is called as excitatory neuron. Now again based on types of the transmitters, they are again labeled. For example, if neurotransmitter formed in this neuron is dopamine, it is called as dopaminergic neuron. If it is forming acetylcholine, it is called as a cholinergic neuron. If it is forming epinephrine, non-epinephrine, adrenergic neuron, like this names are labeled. So it is nothing but a simple cell with all contents required for production of a protein, that is a neurotransmitter. So you can see here also these are the dendrites, which are basically the receiving end of a neuron. This is the axon, which transfers substance from one place to other place in both directions, and this is the nerve ending where it communicates with other neuron. So like this combinations are, it's like two bones are there connected with the joint. So similarly two neurons are connected by the synapse. So here close look, you can see here it is having, cell body is having a nucleus. These are the dendrites. And important thing is that this axon is there, is again of two types. In some, this Schwann cells are present only, and in some the Schwann cell forms multiple layers around the axon, that is called as a myelin sheath. Now based on that again we classify into two categories. So this is again a typical neuron having a cell body, dendrites, axon and nerve ending. So anywhere in the brain you will find similar structure only. There will be no difference of this length of the axon only. It may be very short, it may be very long. Now in the axon, important thing is that there is axonal transport. As I mentioned, this is the typical cell having nucleus, nucleolus, which will contain again DNA, RNA. It will have a Golgi apparatus. It will have endoplasmic reticulum. Typically everything under guidance of the DNA and RNA, there is production of a protein which is called as here neurotransmitter. Now it is formed in the vesicle Now these vesicles are transported from here to the nerve ending for release at the synaptic ending. So this transport away from the cell body is called as the orthograde transport. So there is a transport of vesicle from here. So at the synaptic ending, you might have studied at the neuromuscular junction, there is a release of neurotransmitters. So the vesicles which are filled with the neurotransmitters are coming towards the end and they are releasing it at the ending. So this is called as orthograde transport. And once the transmitter is released, remaining material or excess vesicle goes back towards the cell body for recycling. It is called as retrograde transport. So in axon, transport takes place in both direction, away from the nerve cell body and towards cell body. And in that, what happens when there is retrograde transport, what happens, some of the viruses can come and go towards the brain. So this is again one of the way of transport of virus towards the brain. For example, the rabies virus. Okay. So two types of transport are there in the axon: orthograde transport and retrograde transport. So it will have again the molecular motor we can see in this. For transport, they are having a kinosine protein, molecular microtubules which are helping in the transport processes. Now the glial cells, which are the supporting cells of nervous system, is basically neuron are the basic of nervous system. To support the neuron and to protect the neuron, there are the glial cells, also called as neuroglia. Now they are classified in two categories: small in size called as the microglia, big in size called as macroglia. Now this microglia is nothing but basically a defense system of nervous system. Just like in our blood, we are having WBCs. In the different categories are there. Ultimately, there is formation of macrophages in the WBC once they go in the organ. Similarly, microglia is nothing but a macrophage of nervous system, and it helps in the protecting and removing the debris from the injury, infection and diseases. So it is nothing but a scavenger cell taking part in the defense of brain. It is called as a microglia. Now the macroglia are three types of cells: oligodendrocytes, Schwann cells and astrocytes. These astrocytes are appear like a star, and they are basically supporting the blood vessels and they are forming the blood-brain barrier. And these two cells, oligodendrocytes and Schwann cell, they are basically covering the axons. Covering the axon in the brain and forming myelin sheath are the oligodendrocytes. Covering axon in the periphery are the Schwann cells. So these are the neuroglia. Now let's see more detail regarding this oligodendrocytes and Schwann cells. Now here you can see these are the Schwann cell which are giving sheaths and encircling the axon about almost 100 times and forming a big insulation surrounding it. It plays a very important role to prevent leakage of the current, because it is like acting like an insulator. And they are scattered in the axon along the length, leaving only spaces in between. That space is called as nodes of Ranviers, where they are not covering the axon. So Schwann cells are basically cells which are forming myelin sheath in the peripheral nervous system, and they are surrounding only a single cell, and you can see the layers of myelin sheaths are multiple, around 100. On the contrary, oligodendrocytes are forming the myelin sheath in the brain, that is in the CNS, and in that but you can see a single cell is covering 4 or 5 axons. Single cell is covering multiple axons. Again, it is forming similar myelin sheaths across them only. So oligodendrocytes are forming myelin sheath in the CNS, and Schwann cells are forming myelin sheath in the peripheral nervous system. Now let's study something regarding this myelin sheath. Now based on the presence of myelin sheath, we classify the nerve fibers. First classification based on myelination, myelinated and unmyelinated axons or nerve fiber. Based on the functions, motor and sensory neuron or axons or nerve fiber, and based on diameter and conduction velocity, This is called as Erlanger-Gasser's classification. We'll study later what is this. So these are the three types of nerve fiber. Three classifications are there based on the first is based on myelination, second is based on the function, third is based on the diameter. So in this how we are classifying nerve fibers in different categories. Now based on myelination, myelinated axons. Myelinated axons have covering around their axons called myelin sheath. Myelin sheath is formed by Schwann cells in peripheral nervous system, and as I mentioned, it is forming at least about 100 layers around the axon, and one Schwann cell is covering a single axon. On the contrary, these oligonucleotides are forming the myelin sheath in the CNS, and a single oligonucleotide is covering many axons at a time. And myelin sheath is what is? It is a protein and lipids and water. Cholesterol, phospholipid and glycophospholipids are there. Large diameter somatic nerve fibers as well as preganglionic fibers of autonomic nervous systems are also myelinated. You can see these are the Schwann cells. These are covering. You can see this is the myelin sheath. And at intervals, myelin sheath is absent. That interval is called as nodes of Ranvier. It has got a very important physiological significance. We'll see. Basically, there are, if you just recall, action potential, current, how the current passes or depolarizes. So it is nothing but voltage gated sodium potassium channels across the cell membrane. So basically, the distribution of voltage gated sodium channels across the nerve membrane makes the difference in the conduction. Only 50 to 75 voltage gated channels are there on the cell bodies. Okay, so that's why only production will take place here. Around 350 to 500 in initial segment. Less than 25 on the surface of myelin sheath. Just see how much on the myelin sheath. It is only less than 25. Means it is almost, it cannot conduct impulse across this one. And on the contrary, you can see almost 2000 to 12,000 voltage gated sodium channels per square meter at the nodes of Ranvier. This is the place where conduction is maximum, and 20 to 75 at the axon terminal. Along the axons of unmyelinated neurons, it is only 110. So this is the distribution of sodium voltage gated channels across the neuron. Simply based on this, you can find which area will have a maximum conduction. So obviously, as the number of channels are more, if you recall physiology of action potential, it is nothing but rapid influx of sodium that will lead to depolarization, and efflux of potassium that will lead to depolarization, and that is nothing but action potential. So obviously, more the channels, more will be the action potential generated that side. In many myelinated neurons, the sodium channels are flanked by potassium channels that are involved in repolarization. Now, based on this presence of channel, you can see that the myelin sheath is effective insulator. It will not allow the current to leak outside. Obvious reason is the number of channels are less than 25 here. So current cannot get generated and passed from here. So current flow through it is negligible. So it is acting as an insulator. Depolarization in myelinated axon travels from one node of Ranvier to the next nodes of Ranvier, because obviously there are 2000 to 12,000 sodium channels are there. So current generated is very fast here. So current jumps from one to other. So this is called as the jumping of depolarization from nodes to other node is called as saltatory conduction. It is a rapid process that allows myelinated axon to conduct up to 50 times faster than the fastest unmyelinated fibers. So this is the reason myelinated fibers are having fast conduction velocity as compared to unmyelinated. So more the conduction of impulse fast, obviously more will be the reaction, more will be the excitation contraction coupling and the muscle contraction. If the current is, just in simple way, if current is passing continuously, we'll have a continuous contraction of the muscle as required. If current is passing very slowly, we will have a slow contractions. So based on that, Myelinated have got more conduction velocity as compared to unmyelinated. So here you can see generation of current is fastest here. So immediately transfers from one to other sides. So this is called as saltatory conduction. On the contrary, in unmyelinated axon, which do not have myelin sheath, so they are having only 110 sodium channels. So because of this, generation of impulse is less. So commonly these are the somatic nerve fibers of small diameter, postganglionic fibers of autonomic nervous system, dorsal nerve root nerve fibers in the CNS, and speed of conduction of impulse is slower in this. So this is first classification of nerve fiber. Nerve fiber is nothing but an axon of a neuron. So axon of a neuron is a nerve fiber. So based on the presence of myelin sheath, we have classified in two categories: myelinated axons, unmyelinated axon. Axon is nothing but nerve fiber is nothing but axon of any neuron. Now, second classification of neuron is based on the functions. Now here I have shown one neuron. This is the part of skin or the receptor. So whenever we stimulate the skin or any receptor of the body, impulse generated, it will travel towards the brain. So if it is collecting sensory information from the surrounding to the brain, it is called as sensory neuron. On the contrary, when we are making some action, the neurons in the brain are sending impulse down to the muscle for contraction. So if impulse is going from neuron to the muscle for the movement, it is called as a motor neuron. So based on this, we are classifying neuron in two categories: sensory neuron and motor neuron. Third category are neurons which are connecting the two neurons. These are called as interneurons. Their function is only connecting two neurons. So this is called as interneuron. Now these are the anatomical different types of neurons. Some are unipolar, having only one pole. Some are bipolar, having two poles, like in case of retina. Some are pseudo-unipolar. So these are the anatomical classifications actually. Functionally, we believe in only two: sensory, motor, and third interneuron, and one is based on myelination. So this is the anatomical classification: unipolar, bipolar, and multipolar, which are having multiple connections with the different neuron. You can see like typical neurons in the cerebellum. They are having bunch of connections, thousands of synaptic connections are there with a single neuron. So these are anatomical classification of different neurons. Now, very important thing, the the axons, the neurons are either in the brain and spinal cord, or they are at the receptor. So if you can see, if there is a stimulation here, what will happen? Action potential generated, it will travel along the axon towards the brain. There is a part of sensory. So these are all these are axons are nothing but all these are nerve fiber. What we mentioned, ulnar nerve, radial nerve, axon nerve, is nothing but bunch of axons of the neurons originating in the spinal cord, or some originating in the periphery. If it is a sensory, it is originating in the periphery. If it is a motor, originating in the here. So you can see here. So generation of impulse going towards the periphery of the execution, it's called as a motor nerve fiber. So based on that, based on the axon, again we classify the nerve fiber in sensory nerve fiber, motor nerve fiber. These are the divisions of spinal cord. We are again labeling the nerve based on the section from where it is arising. If it is arising from cervical segment, it is called as a cervical nerve. Arising from thoracic segment, it is called as a thoracic nerve. Arising from the lumbar segment, it is called as a lumbar nerve. Arising from sacral segment, it is called as sacral nerve. Again, to recall, nerve is nothing but axons of a neuron. Now again we classify the nerve fiber based on their origin. If it is arising from the spinal cord, it is called as a spinal nerve. Mind well, it is nothing but again axons of the neuron. These neurons are in the spinal cord. And if it is originating in the cranium or a brain, it is called as a cranial nerve. That means neuron of the brain their nerves are called as cranial nerve. It is again axon of a cranial neuron located in the brain. Again we classify them in sensory fiber and motor fiber. Motor fiber again we classify in two categories. Somatic, it is supplying the skeletal muscle. Autonomic, it is supplying the autonomic organ. And again autonomic, two categories, sympathetic and parasympathetic. Means all these nerve fibers are nothing but axons of the neurons based on their locations. Now this is important classification explained by Erlanger and Gasser. It is based on fiber diameter, and as the diameter decreases, conduction velocity decreases, or in the reverse way, as the diameter increases, conduction velocity increases. So this classification is based on diameter, conduction velocity. Based on that, they are classified in A, B and C. Now A further divided into A alpha, A beta, A gamma, A delta. So gradually there is a decrease in diameter. So A alpha is having a maximum diameter, that is 12 to 20 micrometer is the diameter, and that is having maximum nerve conduction velocity 70 to 120 meters per second, and it is required especially for our motor activity. So that's why somatic motor nerves are having A alpha fiber. Proprioception, joint position, we need to adjust joint position constantly. So this is the A alpha fiber. Now gradually you can see A alpha, A beta, A gamma, A delta. These are the fibers which are carrying the important sensations. So this is called as the Erlanger-Gasser's classification. Different sensation they are carrying: proprioception, touch, pressure, motor impulse to the muscle spindle, then pain, temperature. So this is the division, this is the classification. And important thing is that based on this classification, there is one more criteria on this classification also. The different neurons are sensitive to different external factors. For example, local anesthesia, pressure, some are sensitive to the pressure, some are sensitive to the local anesthesia. Now it is a part on you to learn, and when we come for the lecture we'll discuss which of these neurons are sensitive to, out of this, which are sensitive to pressure, which are sensitive to hypoxia, which are sensitive to the local anesthesia, because it is very important when we are treating a patient or giving local anesthesia, we should know which nerve fiber are sensitive to what. This is one more classification, that is numerical classification. Now it is based on numerical 1A, 1B, like this classification. It is again nothing but same with the difference. Okay. Now these are regarding the classification. So we have classified in three categories. One is based on myelination, second is our classification is based on function, third is based on this diameter. Now functions of the neuron is basically, all basic function is one, generation of action potential and transfer of impulse from one neuron to other neuron. It is only connectivity, a neuron and stimulating other neuron. Ultimately either there is an execution by muscle contraction or granular secretion. So parts of neuron taking part in this one: dendritic zone, which is a receiving end, it receives the inputs from other neuron. Action potential generated at the initial segment of a neuron. Axon is basically for transport. It transmits the propagated action potential towards the nerve ending, and at the nerve ending there is a release of transmitter, and through the transmitter there is generation of action potential in the subsequent neuron. So basically functions of neuron is nothing but generation and transfer of impulse. Axon transmits the action potential from one end to other end, and at the synaptic ending there is a release of neurotransmitter, and next neuron gets either excited or inhibited. Now properties of nerve fiber: excitability, conductivity, refractory period, and all or none response. Now these all four properties are nothing but same properties of action potential which we have discussed in a lecture of action potential and resting membrane potential. Excitability means ability of any cell to generate action potential. Neuron is excitable. Conductivity, it conducts the impulse. So axon is conducting the action potential from one end to other end. Refractory period we have already discussed in detail with the during action potential. What is a refractory period? All in all response is same for the action potential. If you are giving a threshold stimulation, then action potential generated is maximum. With the supra threshold we will not get. Same height will be there. Now few additional properties nerve fiber is having: unfatigability, summation and accommodation. So let's see these three properties only. Unfatigability means nerve fiber cannot be fatigued even when they are stimulated continuously, because this is nerve fiber is conducting impulse without expenditure of any energy. Summation, application of subthreshold stimulus does not evoke an action potential, but if subthreshold stimulus are applied in a rapid succession, they are summated and they produce an action potential. So this is the property of summation. And last property, very important property of accommodation: application of continuous stimuli may decrease the excitability of a nerve fiber, a phenomena called as accommodation. This is very important for our survival. We will discuss this during our FCR discussion. If a nerve is submitted to passage of constant strength of current, the site of stimulation shows decrease in excitability. The accommodation consists of a rise in threshold of a tissue during stimulation. A similar feature observed at the nerve ending is called as adaptation. So this nerve fiber accommodates while the nerve ending adapts. So here we are finishing our talk of. So we will study this during our lecture. So to summarize, we have discussed types of nerve fiber, functions and properties of nerve fiber. So here